ASH 2015 coverage continues. I'm Thomas Baldrick. Always a pleasure to have with us Dr. Ruben Mesa from the Mayo Clinic in Scottsdale, Arizona. Nice to see you.
Nice to see you again.
I know your conference is busy. Let's get to talking about one of your projects, study on the relationship between patient-reported outcomes and health-related quality of life and efficacy in patients with myelofibrosis in your phase 3 Persist-1. What can you tell us about this, just as a background?
Sure. So the Persist-1 study was a pivotal phase 3 study looking at Pacritinib versus best alternative therapy for patients with myelofibrosis who needed therapy and included patients that had marked thrombocytopenia. So it was for the broad spectrum of patients with myelofibrosis, but one of the distinguishers about Pacritinib we had learned from earlier studies is that it was safe to give in patients with thrombocytopenia. Some of them, their platelets even improved. Patients who had anemia, their anemia might improve as well. So we presented the initial data at ASCO, which showed deep activity for splenomegaly and symptoms in the trial, including people with marked thrombocytopenia. What we have here at this meeting is a much deeper dive and a longer data set regarding the exact outcomes in terms of the patient-reported outcomes, how it related with therapy, how it corresponded with response, and how it corresponded with between symptoms and quality of life. As we think about patient-reported outcomes, people sometimes interchange symptoms and quality of life as being the same thing, but they're slightly different. Symptoms are really individual aspects that you notice. Fatigue, pruritus, bone pain, they are quantifiable difficulties. When we think about health-related quality of life, there are many aspects. It's overall, as we add everything together, how are we doing? As well as other factors, stress, uncertainty, even the hassle of medical care. That all boils into that health-related quality of life. In this particular study that we present here, what we looked at was the MPN, or the MF symptom assessment form, a validated instrument used in the trial. We compared it against the response. We compared it against other metrics of quality of life we included in the study. The EORTC-QOL-QC30, which is a well-validated, broader instrument of quality of life, as well as the EQ5D. What we found, one, that there was very strong correlation between symptomatic response and improvement of quality of life. Two, we saw that very specific aspects were important contributors. Reduction in the spleen, improvement in fatigue. They were really tied closely to that patient's perception of improvement in quality of life. Finally, one of the key areas of interest in this study was that patients who had marked thrombocytopenia, platelets of less than 100 or less than 50,000, one had very significant symptomatic responses, but two, this pattern was exactly the same. So we're really achieving what we wanted to achieve. For that sickest group of patients with MF that have severe thrombocytopenia, they now too can get relief from splenomegaly and symptoms in a way that impacts their quality of life with the therapy with Pacritinib.
This has got to feel good because quality of life for these patients is so important.
Absolutely. When we think about a disease like myelofibrosis, it is a chronic disease. And we hope with the impact of therapies, JAK inhibitors, other interventions, that hopefully these patients will live for many years. So the quality of life is very, very important. And the disease impacts them in ways that really severely impacts their quality of life, both in terms of pruritus, itching, bone pain, but it's just kind of a level of discomfort that really causes tremendous suffering, and it's very satisfying to see that we're really alleviating that suffering.
Now, with the Persist trial, you've also got another trial going on alongside of it. What else will you be doing going forward with what you've presented here at ASH?
Well, the Persist-2 study, which is ongoing, along with my colleague Serge Rostovchik and many other colleagues, is a deeper dive in particular for those patients with marked thrombocytopenia. So there the eligibility is less than 100,000 platelets, so everyone is very specific in that group. We're doing many more efforts to, one, quantify those benefits, but two, really taking a deeper dive in terms of the science as to why we're seeing the benefits. There are a whole variety of different inflammatory cytokines that we think are part of all of this benefit. in addition to some of the improvement we've seen with the anemia and thrombocytopenia. Again, when I try to view symptoms, I don't view it as a separate entity altogether from the disease. It's all interrelated. The symptoms, the cytokines, the cytopenias, the splenomegaly, these are all manifestations of really what the clone is doing. There's interesting data that we've previously presented that other cytokines, such as HRQoL and other pathways, might be active in these diseases that are selectively inhibited with a therapy like pacritinib and might be responsible in part for symptom benefit, but also cytopenia improvement.
Is it safe to say the advancements in treating myelofibrosis that just being able to do deeper dives is a good sign for patients?
Without question. I mean, several years ago, there were really no other studies in myelofibrosis other than small, single institution phase 2 studies. It was a trial of 10 patients, 15 patients. I think back to an ASH of 10 years ago, an oral presentation would be 12 patients treated with a drug on a single-arm study. Now we're looking at very deep dives of, very mature randomized data. We saw today the 5-year data from Comfort-2 from Dr. Harrison, where they would do very deep analysis. We saw a deep dive in terms of molecular responses for patients on imetelstat and what they mean in terms of response. So I think, even across the different classes of agents we're seeing, it's at a much, much more sophisticated level, and it's really giving us information to hopefully continue to advance the treatment paradigm further.
Keep up the good work. Always a pleasure.
Thank you very much. Thank you.